Okay, so we'll get started with our 11 o'clock and our next two talks. And so the next talk is Dr. Christine Foreman from Montana State University. Christine's talk is entitled Extreme Environment Microbiology and ISIS. And we'll give it a go.
Thanks, Scott. All right, well, thank you all for coming back after the break. Hopefully everybody's well caffeinated again and ready. When Scott gave me this title, Extreme Environment Microbiology and ISIS, I confessed that I went out and crowdsourced. So I went to a number of people and kind of said, okay, help me out here. So I want to very much acknowledge the help from these folks, but also acknowledge that any mistakes are solely my own. Can everyone hear me? Good, great, thank you. So as a broad overview, the cryosphere includes environments that where our water appears in its frozen state. So lake ice, river ice, snow, ice caps, glaciers, sea ice, permafrost, which is a permanently frozen ground. And while it might be tempting to lump all of these environments together because they're frozen, the cryosphere hosts a huge diversity of environments as well as different physical properties, right? So the temperature ranges moves around, the consistency moves around, the ice morphology is different, the availability of water, the availability of different nutrients, salinity, light, all these kind of things are very different in each of these. So within each of these, though, organisms exist in the porous spaces between the ice. And so I'm going to talk today about some of these different environments. So it turns out that, well, I think that Earth is a great place for icy analog research because from a biological perspective, our Earth is a cold planet, right? So most of the Earth's surface is covered by oceans, which are less than 5 degrees Celsius. About 80% of the terrestrial biosphere is permanently frozen. So understanding how organisms work in these cold and icy areas can help us to better search for life on other planets. I like this graphic because it provides a nice overview of the cryosphere environment on Earth. You can sort of see this exploded and expanded view, and it kind of really sets the stage for what we're going to be talking about. In this area, what we see are areas of significant snow cover, and these are shown here in green. I get my pointer here. Right, so we've got these areas of significant snow cover in green over here. The snow matrix is generally characterized by low temperatures, high ultraviolet radiation flux, and scarce nutrients. And this leads to a relatively low abundance of microbes in snow. These microbes get here because they're transported in the atmosphere and they get deposited as snowfall as they fall to the Earth. Interestingly, snow could arguably be classified as a mineral. So it's solid, it's homogeneous, and it has an ordered arrangement of hydrogen bonds between the water molecules. Because this is a 101 style talk, I've tried to throughout sort of include some papers that might give you some reference to other works that might be of interest as we kind of go through this. So sea ice, shown here on the map in white. So the white area is through there. It covers approximately 25 million square kilometers on our planet, and it's an important habitat for the diversity of life. It forms, grows, and melts in the world's oceans, and it waxes and wanes seasonally, right? So let's see here. We can see the September maxima around Antarctica. This is the largest extent of sea ice coming off from the continent versus the February minima where we lose all of that sea ice. So again, a very dynamic environment that we have changing in here. Sea ice differs from icebergs, lake ice, and glaciers in that the sea ice forms from salty water from those oceans, right? When that sea ice forms, it excludes salts into the water below. So then you create these dense masses of water that then sink to the bottom. And as these sink, then these salts are extruded, you create environments within the sea ice itself, so these porous brine channels that are then created when the salts get excluded. It's also important for albedo, right? When we think about albedo on our planet. So sea ice, if you have sunlight hitting white surfaces, obviously you're reflecting that energy back into space. If you have open ocean, then you're absorbing that. So you have these continuous sort of feedback loops as the sea ice extent moves. So sea ice is interesting as a habitat, right? In that there's an entire microbial cycle occurring within. So the initial colonization occurs here in the autumn. So as ice gets advected to the bottom of the sea ice, it actually brings microbes up with it into the sea ice here. They get entrained into that sea ice, and then they move into the winter period. So in the winter, there's not a lot of activity in there, and so you sort of get things sort of slowing down. And then spring and summer hit, you get this influx of solar radiation, you get a large bloom of organisms, and biomass begins to accumulate. The sea ice algal bloom ends when either they run out of nutrients or that melts, and then you recharge those organisms back into the ocean waters below. Cryobrines are another interesting feature, icy feature, whose high salt content maintains them in an unfrozen state. So conditions at temperatures below 0 degrees Celsius. These can host a diversity of different organisms, and in some cases can be highly productive. One interesting example of these are features known as brinicles. So these are hollow tubes of ice. So you can kind of see here. This is a nice colored version of that. So hollow tubes of ice that form under the floating sea ice. When the dense cold brine exits the sea ice conditions there, you get to the seawater, and that's close to freezing. And so you start the crystallization, and you get the generation of these brinicles forming through here. This schematic from Vance et al. Shows the differences between the surface, the ice, and the ocean water below on Earth, Europa, and Enceladus, and sort of the predicted scaling of these, so the size of that ice, as well as potentially the size of those brinicles within that system. These images here came from Maddie Garner's work in our Sub-Zero laboratory at Montana State. So Maddie's been able to start growing these brinicles in the lab, couple 4 to 5 sort of centimeters down. It's been a challenging, I can say that, challenging experience, but she's able to actually sort of figuring out those conditions. And these brinicles are interesting because they're hypothesized to kind of be a sort of inverse chemical garden within sea ice. These glaciers and ice caps, so shown here in the black and the black dots, so they sort of serve as museums back in time. They offer high temporal resolution of things like temperature, precipitation, different chemistries, microbes, all sort of slowly going back into time there. The schematic on the right shows what happens when snow falls onto the surface of that ice sheet, right? So you start with these nice sized crystals at the beginning. As more and more crystals land and snow compresses, you start to change and transition into granular ice, firn, and then the final glacial ice. And what this transformation does is it starts to remove the amount of air within that space. You compress it, and eventually when you get to glacier ice, you've got these sealed off bubbles of air that were trapped at the time that everything sort of sealed. So you can look back and say, okay, 300,000 years ago, here was the temperature within our system. Here's what the chemistry of the atmosphere looked like, and also trap other things within these bubbles. So glacial ice and snow are very much attuned because snow starts that process to make the glacial ice as it kind of goes through there. The glacier ecosystem, or ice sheet ecosystem, can be divided into 3 broad realms. The supraglacial environment, so that top surface here that we see, you can sort of see a cryo lake at the top there. End glacial system, so this is the majority of ice on an ice sheet or an ice cap, is this end glacial environment through here. And then the subglacial environment, right? And that's occurring beneath the glacier or the ice. So of these 3 environments, the largest one is the one that we know least about, right? So there's lots of studies in the literature about the supraglacial environment, features on these surfaces called criconite holes, and the subglacial environment. There's some really fantastic work looking at those. But there's this whole other environment here that we know very little about, and that tends to be very nutrient deplete, as well as deplete in microbial cells. This cartoon of glacial ecology shows these three realms. So if you start at the top, right, so you get that superglacial environment here, and what you have is sunlight fuels the carbon fixation by algae, cyanobacteria. It's then used by heterotrophic bacteria, so this sort of nice tight coupling. Sometimes you also get organic matter deposited on the surface of that glacier, so it's sort of that dark material that's landing, which has an effect on the albedo of the system. It begins melting in to form those sort of small criconite holes on the glacial surface. And with these holes, microbes are attached to the individual sediment particles, forming a biofilm around them and a sort of hotspot for microbial activity. As the cracks form throughout this endglacial environment, you can get movement of water and other nutrients, materials that get transferred from this sort of surface down through the endglacial into that subglacial realm. And we move from primarily a photosynthetically dominated environment down to a chemolithoautotrophic environment within the system. So again, the idea I want to get across is that all ice is not the same, right? There's very different sort of transition zones, different things that are occurring, but yet still quite a bit of connectivity between these in some ways. Let's see here. Okay. In the superglacial environment, pigmented organisms occupy the glacier surface niches and also impact albedo just the same as it did with the sea ice. The caption on the left, so the schematic here, is from our work in 2016, where we really looked at the close coupling of microbes within one of these criconite holes. So if you can look at this, you can kind of envision the schematic. So you've got these holes that come about on the ablation zones of glaciers. So they start when this dark material hits that glacier surface, the sun warms them, they melt into the glacier surface, and you have this extremely active, in many cases, sort of mini-entombed biofilm on the glacial surface. So you've got lots of things being shared, nutrients being cycled and moved throughout this environment. We also see that microbes from these superglacial environments often have a huge range of pigments associated with them. So when we isolate them or enrich from them from the environment, we see this beautiful rainbow of different pigments that are associated, and they give some survival advantage to organisms. They help with radiation tolerance, right, acting as nice sunscreens. They also help in terms of membrane fluidity by giving more fluidity to those membranes there. So they're a common feature when you're isolating organisms from these environments. This image from Alex Inicio's group also shows nicely what the different sort of pigmented environments are on the Greenland ice sheet. So you can see the nice red snow, algae, cyanobacterial mats, and things like that. So superglacial features have a lot of pigmented organisms that act as nice signatures for us. After snow falls again on the glacier, it's compacted into those porous layers called firn. You get some transition of gases and things moving around within that firn layer before it eventually becomes sealed into the glacial ice itself. So these ice cores, typically they're about 10 centimeters in diameter, and they can go down to kilometers, right? So in the Antarctic, you might get a 3 to 4 kilometer sort of ice core going down into there. And here you can see the really nice dust and debris layer, so probably volcanic ash. This image came from the West Antarctic Ice Sheet Divide project. And this schematic on the right was one taken from our cryostage microscope. And so what you can see are individual ice crystals. And where they come together, you have these veins and triple junctions, and that's where the bacteria exist. So as you go through and you sort of freeze thaw ice, what you're doing is you're pushing the bacteria into this, and you've got this thin film of water, right? And that's your environment within there. This whole sort of theory was postulated first by Buford Price in his article in PNAS, and he sort of hypothesized that this was how you were going to have these triple junctions coming together in these ice veins, and how you'd have a nice microbial habitat present because you have this thin film of water within these cages. As I mentioned before, ice cores preserve microbes along with the more typically measured chemical components within these. A few years ago, we examined ice from Bird Station in Antarctica, so about 21,000 years before present, so in a period called the last glacial maximum, as well as ice from the West Antarctic Divide ice sheet, and this was about 16,000 years before present during the last deglaciation. And what we can see is that there were differences related to the climate and the environment at the time that this ice was deposited. So we see the bioavailability decreasing as you move into the younger ice. We see differences in the microbes that can be recovered from those different ices. And so we can kind of look at this because the conditions were different. So in the last glacial maximum, the cartoon's supposed to show that this is the ice cover, right? It's pretty continuous. There's lots more wind. There's lots more things that are being deposited onto that ice sheet as well. Versus during the last deglaciation, you kind of see a breaking up of that ice cover, less wind, less things being deposited. And so we can sort of relate that back to the environment in which the ice came deposited. Interestingly, when we recover a lot of these organisms from deep glacial ice, they're tiny, right? This is work from Mateva and Brenchley showing these really tiny ultra micro bacteria. And so when we think about looking for life, that posits a challenge, right? So these organisms are pretty nutrient starved, but I think back to one of the things that Karen mentioned in sort of like time, right? So time to us is very different than geological, I'm not a geologist, but like geological time, right? And so thinking about what it takes in order to resuscitate an organism that's that tiny and pretty much is only repairing its DNA for the most part. You know, maybe its replication time might be 200 years. I'm just spitballing, throwing wild numbers out there. Right, it could be something along that line. I wouldn't think, Karen, that those are the super fast ones, but, but you know, the time scale is very different for us, and just sort of thinking about what that might look like, it poses a problem. And when we start thinking about where we're going to find these organisms, especially in end glacial ice, which it's pretty scarce, those are tiny sort of shots for us to be going for. There's also a large diversity of subglacial environments on our planet that we can explore. So a few examples include the hypersaline lakes in Devon Island, in Canada, below the volcanic calderas. This subglacial lake exists in Iceland. And there's over 400 subglacial lakes that are known at this point below the Antarctic ice sheet. So essentially the Antarctic ice sheet covers this massive amount of water and water features, potentially wetlands, all kinds of different features that we're just not aware of because it's under about 2 miles of ice. Jill McCookie and others have been investigating a really fascinating subglacial feature known as blood falls. And so on the left, you can see this really striking view of blood falls as it's discharging from, it's a conduit from below the Taylor Glacier in Antarctica. And episodically, it's triggered to produce this brilliant hue that comes out of the glacier there. And the inset up here is of organisms that Jill's lab has cultured from blood falls. And again, you're noticing these really nice pigmented organisms as well. This schematic on the right explains the conditions and the resident bacteria that can be found within blood falls. And again, super fascinating to think about what triggers this sort of output. And for questions about that, you're going to have to talk to Jill, because this is way outside of my area. But I just think it's a really amazing system when we start to think about these subglacial conduits to the surface and how we can sample them and how we know when to sample them, right? So we can't just always go there and collect a sample and assume that it's going to be from under Taylor Glacier, right? So there's a lot of geochemistry that really determines when you're sampling the subglacial water versus just the glacial melt that comes from that. And I'm sure throughout we'll get more information about that. So how is it that microbes can survive in these cold and often inhospitable environments? So low temperatures have multiple impacts on water availability, thermal energy of reaction rates, metabolic processes, molecular stability. So in addition to temperature, there's also a lot of other stressors that come together, sort of have this intersectionality in cold temperature environments, right? So things like low nutrients again, potentially high levels of salinity, desiccation, high levels of ultraviolet radiation, and oxidative stress, because oxygen becomes more soluble at colder temperatures, so you get this reactive oxygen stress that happens too. So studies have shown that these cold temperature organisms are not necessarily endemic, right? But instead, they're globally distributed. What's different in them is the adaptations that they have for life, right? So you're not going to find an organism, well, I'm not going to say that, I guess. You're typically probably not going to find an organism that's only found in Antarctica or only found in Sahama in Bolivia or in Tibet or things like that. The general sort of clades and lineages that we see across the cryosphere. But it's those adaptations that allow them to happen. Because everything's transported all around. Not everything is going to exist when it lands in a cryospheric environment. And so organisms can use strategies at sort of all different levels of cellular physiology to combat these types of stresses. So characteristics of cold adaptation include the expression of cold shock proteins, EPS, the presence of compatible solutes, things like glycine, glycerol, trehalose. So those help reduce the freezing point of the cytoplasm within those cells. They help to scavenge those free radicals and to help to stabilize those cellular membranes. So there are also structural changes to enzymes, to the cell envelope. So lots of different ways that organisms have adapted to live in these cold temperature environments. So when we look at that and we think about the cryosphere, you might think that these environments are lacking in life, but it's really far from the case, though Scott would probably argue. So the cryosphere microbiota has an unexpected diversity, so with large genetic as well as functional potential and diversity. So these are summarized nicely in a paper by Anja Boschis, Jill McCookie, and others from 2015. So if you haven't seen it, it's really a great paper. And so we can kind of look at these two environments, right? So we've got the supra and endglacial kind of thing up here going on. And so the top panel shows that these organisms in the superglacial environment are dominated by photosynthetic microorganisms. So cyanobacteria, the really brightly covered snow, ice, and algae, and heterotrophic bacteria. And then in those subglacial environments down below here, we really have domination by the chemolithoautotrophic communities. So they use the minerals and the carbon substrates that are found to generate energy from redox reactions. So the bottom panel then shows sort of the same sort of thing that I've showed earlier with what's happening in snow and ice. I just think this is a really great article to highlight for everyone. From a general point of view, two factors most likely define limits for life at low temperatures. One is the lack of liquid water. So if solvent conditions can't be maintained. And two, vitrification, so the temperature at which the cytoplasm transitions to a non-crystalline amorphous solid. To date, there's no generally accepted lower limit of life, so including intrinsic enzyme properties and organisms that can potentially survive as low as temperatures down to minus 196, though that study hasn't been, the study has been reproduced, and I'm not sure how much validity people are ascribing to it these days. But again, the idea being is we don't know what that lower temperature limit, that lower temperature limit of life might look like within these systems. And so when we look at this schematic that we pulled together, So we can kind of see, based upon metabolic activity, so these sort of teal circles here, these are different studies in the literature showing metabolic activity down to those temperatures. And again, this minus 196 is the lowest one that's been published in the literature. The blue represent studies of intracellular vitrification, and the green would be enzyme activity down to about minus 100. We look then, so these are the ranges for Earth. So the low temperature range on Earth, the lowest temperature recorded on our planet was at Vostok Station, so minus 98 degrees Celsius. So we can kind of extend where that temperature range is for Earth. And then we can kind of think about how on Earth, on these ice sheets, the temperature gets warmer as you get closer to the bottom, right? So you start off with that surface temperature, and it's, say, minus 50 or minus 60 degrees Celsius, potentially in East Antarctica, really cold. But as you get towards the bottom of that, you've got much warmer temperatures. You might be down to about negative 10 degrees Celsius. And so perhaps, and this is a big perhaps, this sort of thing could be happening on other icy moons and planetary bodies as well, where as you get to the bottom of that ice layer, you get towards conditions that are more amenable to life, at least as we currently know it. So understanding how life survives in extreme icy environments is important, but obviously can also help in our search for life beyond this. So we were sort of hypothesizing that life can exist in these sort of interstitial veins, similar to what I showed you with Bryce's cage here. And so you've got these triple junctions. And so you can imagine interactions between microbes, between different nutrients, energy sources, things that are going on here. You've got some nice plume ejecta where you could have substrates. And so just sort of thinking about what this could look like as we move through. There are many potential bioindicators that can be assessed and preserved. So I'm going to touch on a few of these next. So one might be changes to chemical equilibrium in the form of astrobiologically relevant gases. So these can be found in ice cores. This is a figure from gas records of Lake Vostok, the Vostok ice core, going back about 400,000 years. We can see these nice records that we can find trapped within there. And to be a good indicator, those gases need to be produced biotically and accumulate in the atmosphere. So in this case, then we get them recorded within the ice. Ed Brooks' lab at Oregon State University has shown some really nice work looking at gases, but he's also recorded really anomalously high levels of methane within these cores that's associated with dust levels, right? And so an extreme example of dust within an ice core is here, a picture of a glacier, of an ice core from the Ong Valley in Antarctica. This core and similar ones have been dated to between 4 and 5 million years old. And you can still recover phylogenetic information from this ice core. And so the idea being that dust is a really nice way to preserve some of this information, and we can record gases within ice cores. Another possible bioindicator are biofilms. So biofilms form when single microorganisms attach to a hydrated surface. They undergo a lifestyle switch, giving up life as a single cell to become embedded in an EPS matrix and form this biofilm. So they can be formed all around us, right? If you didn't brush your teeth this morning, you got some biofilm hanging out there. If you're slipping and sliding across a stream, you're sliding on some biofilm. But why should we care? Well, because these biofilms can not only attach to different surfaces, but they provide a survival advantage to organisms living on their own. So this figure up here shows what happens when you take a cell culture and you repeatedly freeze and thaw it over time. So the stress of that freeze-thaw caused them to overproduce biofilm. This sort of red diaphanous looking material, that's the matrix or the EPS that's formed by the microbes, and those green dots are all the bacteria. So to protect themselves from that physical stress, they overproduce that biofilm. You can also see this is a single sediment grain from one of those crikonites. So these are the criconite holes that you might find on glacier surfaces. And so you can see some really nice autofluorescent cyanobacteria, bacteria, which is the green stained organisms here. And so in this case, for this experiment, in this study, we found about 35% of each sediment grain was covered by biotic material. Other microbial bioindicators, pigments. So I've already shown you this slide, but we can use hyperspectral imaging to then really zoom in on the individual pigments. So this is an isolate of ours called Hymenobacter antarcticus, and you can see the really nice differentiation in pigments when you're looking at it under the microscope. Additionally, many cold temperature organisms possess a lot of different other pigments, so not just carotenoids, but chlorophyll, bacteria chlorophyll, things like that. And we can detect those with some of our field instruments as well. This is a picture of Maddie Willis on the Geno ice fields last year. She's holding some nice snow algae and some microbial mats, and you can get a good signature of that with Raman. So when we look at these sort of things, like if we were a rover moving around, I think it would be pretty obvious to see a microbial mat. It would be pretty obvious to see this. The challenge starts to come when we're looking more at those more sparsely distributed sort of areas of biomass, individual cells. What do we know we're looking at, and what kinds of things can we start to find as well from those? Another indicator would be metabolic activity when we're looking, currently at icy environments. And so we can often discern this using stable and radioactive isotopes at the bulk level. So you kind of melt down something and you throw substrates at it and you measure those. But we can also do this at the single cell level, right? So moving beyond where it might be obvious to where we're looking for this life. And on the single cell level, we can do things like add stable isotopes and follow individual microbes as they take up the different, in this case, 13C labeled bicarbonate or 15N ammonium, right? So what you get using the HISH nanosims, so halogen in situ hybridization and nanosims, is the ability to discern the individual organism and what it is that they're taking up and what they're doing. And so again, just starting to move further down that pathway of looking for these sort of sparse sort of cells. Examining isotope ratios can also tell us a lot about the age and origin. So this was a study that we did looking at carbon transformation on a superglacial stream in Antarctica. And so by looking at isotope ratios, you can see the different sources and transformation of carbon within that system. I'm speeding up a little bit here because I think I only have 3 minutes. So another potential microbial bioindicator would be the biofabric, so the fabric of the ice itself. So many organisms in icy environments produce ice-binding proteins, and there's a whole sort of different array of those. This panel here is from Jim Raymond's work looking at a cyanobacteria nostoc from the dry valleys, and you can see this characteristic pitting and these changes in the signature. This schematic over here shows the whole range of those different sort of ice-binding proteins. Some of them are antifreeze proteins. Some actually change the structure around them and put a sort of thin film around the cell itself. Some allow them to attach to an ice surface. Some nucleate the ice and change that structure. So lots of different strategies. But at what level can we actually start to discern these, right? Thank you. So at what level can we start to discern these? And can we then start to detect them more remotely? What happens when the microbes die? Does this fabric still maintain its shape, or does it have to be developed only in the presence of actively producing these individual compounds? So I'll end with a few questions that Scott posed when he gave me the title and talks like that. One of them was, what do we know about the survivability of life in ice that can help guide our life detection? And I hope that I've addressed that question throughout the talk, but I also wanted to highlight a couple of other key things here. So numerous studies have found that cell concentrations in ice cores are highest in regions of high dust concentration, right? So those things might be volcanic ash, aeolian deposits, black carbon. So example, in Smith et al., we showed that biofilms develop on these cricodite particles and aggregate. Merrick Stiebel's group found that in the Arctic that you could best predict the levels of cellular biomass by looking at the amount of dust in the core. The same thing by Yao et al. In the Tibetan glaciers. And so typical microbial cell concentrations in the Greenland ice sheet, 10 to the 3, 10 to the 5 cells per mil when you're looking at clean ice, clear ice, and anywhere from 10 to the 7th and above when you're looking at that really silty, sort of dirty ice in that system. Scott's second question, what's impacted and improved the degree of those signatures? So as I mentioned before, right, ice really acts as a nice museum back in time for us. And so we can go back really far into the record at this point. So in Beacon Valley, there were some studies showing radioactive isotope uptake back to about 8 million years, so Beidl's work. We can also go back into that Ong Valley core that I showed you. And so again, we can pull back phylogenetic information and potentially activity from these cores. And what are best practices in the design of experiments for assessing cellular communities, right? So when we're looking at ice cores, the best place that you take an ice core from is the place that you're going to get the highest temporal resolution, right? So the ability to kind of get annual layers is like that gold standard. So you're going to go to ice divides, you're going to go to ice domes, you're going to go to those places where you can get that temporal resolution. But then a big thing in ice core work is decontamination. So this graphic came from our work, Kirshner et al. In Icarus, where we found that if you remove the external surfaces on those ice cores, you then decrease the amount of contamination to a point where you feel comfortable that you're actually looking at what was trapped in that ice. Because these ice cores are handled, they've got drilling fluids, there's all kinds of things that are going on with them. And so we intentionally painted the outside of these cores with all kinds of stuff, and we're able to watch those values decrease as we sort of moved our way into those, so that by manually decreasing that outer bit, we felt confident that what we were looking at wasn't a result of our handling. Other important considerations are true abiotic controls. Many true abiotic controls, especially when you're looking at the limits of detection being so low. There's so much noise and things that you really have to have a lot of abiotic controls for there. Microscopy is incredibly valuable for us. Cell counts, right, Maddie? So really looking at the microscopy and seeing that the context, like Frank mentioned, the context of what it is that you're looking at. And when do we consider microbes to be alive, right? So we've got things like they go through dormancy. They're viable but not culturable. At what stage are we getting them when they're half starved in these ice cores and they're only half a micron in diameter? So you want to have we want to have multiple lines of evidence showing us what we believe to be activity within these cores, as well as really clean abiotic controls for what we're doing. And sometimes we need to extend our time range, right? Our graduate students are only around for about 4 years, but sometimes those experiments need to go longer. Sometimes we need to amend things with minerals or vitamins or other nutrients to really determine, can we say if something has an activity within it or not? So a couple of questions to consider. What's the longevity of microbes while frozen? Lowest temperature metabolic activity is possible. Which life detection approaches lead unequivocal results? And is there metabolism in solid ice? So I will move on because I'm getting the high sign. So I thank, again, all the people who helped me, and I'll take any questions.